Welcome, Mindsetters, to this session of revision for matrics. Guys, I hope you're excited. I hope you're ready for this because, guys, we're going to be going through a lot. So you guys need to pay attention. It's revision for you guys for life science. And I've got Aslam here who's going to be taking us through today's session. Aslam, what are we going to be doing today?
Ty, we're going to be looking at paper 1 revision. We're going to try and get through as much as we can in paper 1 today.
All right, awesome. So while Aslam makes his way across to the board, I'm going to let you guys know, guys, make sure you get to me on the Facebook page, www.facebook.com/learnextra, and get chatting to me. Guys, if you're lost anywhere, if you need help, guys, please do not feel ashamed to ask me. But guys, also, I want to stress that you guys have been complaining to us, oh no, an hour's not enough. So today, we've actually made the session an hour and a half. So you guys have more than enough time to get those questions in. Guys. Also, guys, I'm going to be giving away these books. Awesome books for you guys. Revision on life science, maths literacy. There's all kinds of stuff here for you guys. And make sure, guys, I'm going to be giving away 5 of these books to the first 5 callers. And guys, make sure you call 0861 006463. And make sure you call us with a question though. But right now, I'm going to actually hand over to Aslam. Aslam, we're good to go?
Good.
All right.
Hi guys, and welcome to Mindset Live. And we are looking at life science paper 1. Before we actually get into the thick of things, let's look at what paper 1 entails. If we look at it, you'll see that the first part of the work is tissues, cells, and molecular studies. And in that, we look at the code for life. What do we talk about in the code of life? We obviously look at DNA, replication, RNA, protein synthesis. We then move on to meiosis, from there to genetics, and from there to evolution. Those are the sections that you need to know in this section. There you have it. DNA replication, DNA profiling, protein synthesis, gene mutations, and this you'll notice gene mutations comes across throughout our syllabus. From this section right up to the end of paper 1, you will hear about mutations. So we need to focus on that in a lot of ways. We also look at the sequences in DNA. We move on to meiosis. And there we look at the process of meiosis. We look at the significance of meiosis. We look at problems in meiosis. And we also look at the differences between mitosis and meiosis. So there you're going to have to start scratching your heads, guys, because mitosis you did in grade 10. But you need to know the differences between mitosis and meiosis in grade 12. We move on to genetics. This section, guys, starts off with a whole lot of terminology. If you learn the terminology properly, the rest of the sections become very easy. So you need to learn the terminology first. After that, you get stuck into solving problems. And the best way to learn how to solve problems is to solve the problems. Yes, solve as many problems as you can. In that way, you get used to how to set out your answers and how to answer the questions. You'll notice there's a distinct pattern in the way questions are presented, and if you attempt many of those questions, you'll get the hang of it as you go along. A little bit further in our LO3, in other words, how life science is associated with your everyday life and environment and society at large. We go down further there. We talk about genetic engineering, how people can alter genes in organisms to make better organisms. Is this a good thing? Is this a bad thing? Etc. And we talk about GMOs, genetically modified organisms, etc. We then move to the second part of the work, and this is what we call diversity, change, and continuity. And in this section, we talk about evolution. This is a section on its own. It's 40% of your paper. Whatever we've done before this, up to genetics, is 60% of your paper. So obviously, 40% is a lot of marks, guys. And you need to know this work. The first part of evolution, we look at different scientists and their contribution to evolution. The two Darwin's, that is Erasmus Darwin and Charles Darwin, Alfred Wallace, and Lamarck. We look at their theories. And then we move on to natural selection, artificial selection, and comparison between the two. We then move on to speciation. What is a species? How does speciation occur? What's the difference between allopatric and sympatric speciation? And reproductive isolatory mechanisms. We then move on to the evidence of evolution. In other words, scientists that present the theory of evolution, what evidence do they present to us to say that evolution has taken place and that this theory is correct? We move on to the most difficult of this section and the most controversial, I might add, and that is the section on human evolution. And here we're looking at interpretation of phylogenetic trees to show the place of hominids and in the animal kingdom. Characteristics that we share with other primates, characteristics that make us different from other primates, etc. Are we clear on those people? Are we? Yeah, it's in my battery, and I put the battery in. Sorry about that, guys.
So, guys, we're going to take a quick little break, and we guys, we'll be back just after this. See you soon. Welcome back, Mindsetters. I hope you had a nice little break. You went and did whatever you needed to do that was going to distract you, so you have to stick around. Make sure you've gotten a snack, water, anything that you need to stop you from getting up again. So guys, pay attention. Pens, pads out. Start making notes, yeah? Right. But right now I'm going to hand it over to Aslam. Aslam, take it away.
Thanks, Ty. Guys, we go straight into question time, and we start off with the first question, question 1. And this question deals with a certain type of nucleic acid. The question says the diagram below represents a part of a nucleic acid molecule. Study the diagram and answer the questions that follow. Very important to read the introduction to a question, because within this introduction to the question may be very important information that's going to lead you into this question. So always remember that you've got to read what's in the question first. So they've given you a diagram. Here's the diagram here, and straight into the questions. And name the process which this molecule copies itself. So they look at this molecule, and obviously here again, if you read the instructions, you read the questions, sometimes you will get some of the answers as well. They're asking you, when does this molecule make a copy of itself? Or what is the process called? Obviously, the only molecule that copies itself is DNA. RNA does not make a copy of itself. RNA gets a copy from DNA. So DNA, obviously this molecule then is DNA, and the answer here, name the process during which this molecule makes a copy, would obviously be DNA replication. Now, we could also obviously ask in this particular question, firstly, name this molecule. Then obviously your answer would have been DNA. Coming back to where we are with this question now, asking you the question what process is represented here. So obviously the question then would be, name this molecule. And the next question could be is label 1, 2, and 3. Let's look at these things carefully. 1 there, 2 there, and 3 there. Now the clues are given. First of all, this is obviously the backbone of the molecule, and we all know that the backbone of the DNA molecule is phosphate. Yep. So that would be a phosphate. Good. Number 2, this is the question that you've got to be careful about. 2 things that you've got to be very careful about here. In this particular case, it's a bit easy, because on your left-hand side, you see the letter G, which represents guanine. So obviously the molecule or the nitrogenous base that's going to combine with guanine would be cytosine, C. In that case, it's easy. Sometimes what the examiners do, they give you a G on the top, they give you another here at the bottom. Look at this one here. We could have put 2 here. Now, there's no G on this side. However, look at the shape of this molecule here where they got G, and look at this shape of this molecule. Obviously, the shapes are the same, so this would be G, so it would then make sense that this is C as well. So you've got to be very sharp when you're dealing with that type of a question. Okay? And our 3 here, obviously, here's it classic. Here's the A given here. There's the shape there. This shape and this shape are the same, so this would be T. I always tell my learners, AT, at. Adenine always combines with thymine. And cute girl, cytosine will combine with guanine. The other types of questions that they would ask you here is, what is the significance of the bond that is found between the nitrogenous bases? And here, you would obviously say, first of all, what are these bonds? They are weak hydrogen bonds. And what is the significance of this? Because they are weak, they can break easily, so that replication can take place, and they would reform after that. That is the other question there. Further than this, we can also still ask other questions in terms of what is the significance of this process of DNA replication. And here, guys, we then talk about why. The significance always means, why does this process actually take place? And in this case, DNA makes a copy of itself because, yes, you got it right, it makes a copy of itself because we want the daughter cells to have the same DNA as the parent cells. For example, you clip your nails, or you cut your hair, or I may trim my beard very often. Obviously, when the beard or the nails or the hair regrows, we don't find nails growing on my beard. That would be awesome. Or should I say, it should be frightening for some people. Imagine nails growing on my beard, okay? Or hair growing on my nails. That doesn't happen. So what actually happens? When we cut our nails, nail grows back there. When we cut our hair, hair grows. When we get hurt on our skin, skin regrows. Why? Because DNA replication has taken place, and this has ensured that the cell that is replacing the worn out or damaged cell is exactly the same as the cell that it is replacing. So those are important factors that we need to take into account when we're talking about DNA. Now, guys, the whole section on DNA is actually very, very easy. You need to know the structure. You need to be able to draw the diagram. You need to be able to label the diagram when you got it. And you move from there. Let's look at this question again. Tai, you're going to have to take a break there. Tai, can you take over? You take fine.
All right. Guys, right now, I think what we need to do right now, Mr. Aslam, I think we're going to actually take a little bit of a break, make sure that these guys get their heads going, make sure that these guys are ready to pay attention and ready to make more notes. But for now, guys, you guys can go ahead and grab a little break, because we'll see you after this. Welcome, Mindsetters. I hope you had a nice little break. But guys, I can't stress this enough. We're also looking for you guys to call us. Besides just posting questions, guys, feel free to call us on 0861 006463. Put that down, 0861 006463. Guys, get chatting to us because we want to hear from you live. So guys, make sure when you call, and guys, post those questions so I can get them to Aslam so he can help you guys out. But for me, I'm going to hand over to Aslam. Aslam, we're good to go?
Thanks, Tai. We are ready to go. And don't forget, you post your questions. It will always be easier with your questions. We can then obviously help you out in a better way. Here we are just taking sample questions. We are now busy with question 2. And here again, the instructions are very clear. The following 3 diagrams, A, B, and C, show an important process that occurs just before cell division. There's your clue in that question already. It says something that occurs just before cell division. So they give you the three diagrams, diagram A, diagram B, diagram C. Say, name the process that is being shown in the diagram. Let's look at the diagram. Something is happening here, and there was one strand here. At the end progression, we get two strands that are there. So what process is this? Obviously, guys, this is DNA replication. This is the actual. Earlier, we just named the process. Now, in this particular diagram, and this question on this occasion serves two purposes. This is why we've selected this question. One is that it's a question and you're learning how to answer a question. And number two, by looking at this diagram, this explains the process of replication. So in that way, you're benefiting at the same time. The second question says, provide a brief explanation for what is being shown in each of the diagrams A, B, and C. So let's go back to the diagram. So that's A, B, and C. And here, guys, look at these diagrams. And I always tell my learners, it is better to learn from a diagram than to learn from chunks and chunks and chunks of text. This diagram, actually, if you can learn this diagram off, this diagram gives you the whole process of replication. And what I tell my learners is learn simple words when you see these diagrams. Give these diagrams your own names. Look at this one here. This one here, we'll just say unwind. Here, we can talk about unzip. And here we talk about rezip. Three simple words. And yet, they speak volumes. Let's look at each of these words. In the first diagram, diagram A, we can see clearly that the DNA molecule, which is normally spiral, double helix, is unwinding. So the spiral is undoing itself, as you can see here. And as this is happening, we're having the nitrogenous bases moving apart. Unfortunately, I don't have a jacket on, otherwise I would have demonstrated to you what we mean by unzip. In other words, the parts of the zip are moving apart. So the nitrogenous bases are splitting and they're moving apart. And while that is happening, what is happening at the bottom here? We find that for each of these two strands, there are nitrogenous bases that are reattaching as we're moving along. And this is what we call a rezip. In other words, there are free nucleotides in the nucleoplasm that attach to each of the two templates. And lastly, we'll find that we're having what we call the rewind. Can you see at the bottom? It is recoiling itself. So from one molecule of DNA, we end up with two molecules of DNA that are exactly the same as the original DNA molecule. So the first thing that happens is the unwinding, and each of the two strands act as templates onto which two new strands are formed. And these two new strands separate and they rewind. All of these reactions are controlled by enzymes. Now look at the steps. You're starting again, unwind, unzip, rezip, rewind. And your last word is enzymes. So 5 words explains this whole process. We're still with question 2. And this one here requires a little bit more reading, guys. This is analysis of DNA samples from a patient with an illness. So a patient has an illness. Showed that there were 2 different types of DNA present. There were 2 different types of DNA present. One was double-stranded human DNA, and the other was single-stranded virus DNA. The 2 types of DNA were isolated and put into separate test tubes. The analysis of the nitrogenous base composition of each test tube is shown in the table below. You'll notice what I've done with the highlighter, I've underlined the key words. And this is what you need to get used to. Initially, you'll see you'll have to do it physically, But as you use this process, you'll find that you can do it in your mind. You will pick out those important keywords. Let's look at the table. The test tube 1 gives you nitrogenous base composition. So test tube 1, adenine is 22.1%, cytosine 27.9%, guanine 27.9%, and thymine 22.1%. Test tube 2, 31.1 for adenine, 31.3 for cytosine, 18.7 for guanine, and 18.9 for thymine. And the question, which test tube 1 or 2 contains virus DNA? Now, again, remember what I said. We have to look at what's on the top here, the table, and the question to answer this question. So they want to know which one is virus DNA. And let's go back here and let's see what they said about virus DNA. What do they say here? They said that the virus DNA was single-stranded. Very important point there. And that the human DNA was double-stranded. Now, if you know your DNA, you'll know that a double-stranded DNA, for every adenine, there would be a thymine. And for every cute, there would be a girl. For every cytosine, there would be a guanine. The percentages would be the same. So let's look at our table. Look at test tube 1. Adenine, 22.1%. Thymine, 22.1%. Cytosine, 27.9%. Guanine, 27.9%. Let's look at the other test tube. 31.1 for adenine, and thymine is 18.9. Are they the same? Nope. So therefore, which one will contain the viral DNA? It will be test tube 2. All right, they didn't ask you to explain yet, but obviously in life sciences, what I like about it, you can't just guess your way. You may get that answer right, but immediately thereafter, this is the pattern. They're going to ask you, explain your answer in 2.1 in this case. So you've got to know what you're talking about. And in this case here, why did you say that it is viral DNA? And I've explained that earlier, the viral DNA in test tube 2. Why? Because the adenine and thymine base composition is not the same, suggesting that the amount of thymine and the amount of adenine is not the same, so therefore it is highly possible that this is a single strand. Whereas, or, by the way, that could be one answer, or you could have given the reverse of that and said, no, it can't be, viral DNA is test tube 2, because in test tube 1, the amount of adenine is equal to the number of thymine, therefore the contents in test tube 2 was probably a double-stranded molecule. I'm hoping you're with me with that one. And that's it for question 2. We move on to question 3. My eyesight is terrible.
All right, Mr. Aslam is loading up question 3. Guys, make sure you guys keep on posting on the page. I'm not getting enough posts, guys. It's too quiet. And guys, this is matric revision, so come on. I know there's more than 5 of you who want questions answered. And guys, make sure you guys call us. The actual number is 086-105-8262. Make sure you guys call us even. Guys, make sure you get posting. If you don't post, I can't help you guys. Mr. Aslam, do you think we should go for a little break while these guys sort themselves out? Yeah?
Yep.
All right. So, guys, we're going to take a little break, and we'll see you after this. Welcome back, Mindsetters. I hope you had a nice little break. You guys don't have to fidget or do anything else because you went to the bathroom and did all the other things that were going to distract you so you guys can pay attention and write those notes. But by the way, Mr. Aslam, we actually had one of the students, Samuel, who had a question. He says he's kind of lost between the difference of translation and transcription. So I don't know if you can help him out with that.
Good. Thanks for that. I think
No
that
problem.
would actually be a great start to what we are going to do now. We're moving on to questions on protein synthesis. So some of your questions, Samuel, would be answered as we're going along. But suffice to say, let's look at the English words, transcription and translation. Transcription means to transcribe. Transcribe, if your teacher tells you, and he wants to be fancy, if you're looking into somebody else's work, he says, Samuel, stop transcribing from Tabojo's notes. What is he actually telling you? He's telling you, Samuel, stop copying. So transcription is the process of making a copy of something. In this case, when messenger RNA makes a copy of DNA, that's transcription. Translation, to translate, If I start talking here in Arabic or in Urdu or in Chinese for that matter, you wouldn't understand what I'm saying. We would need Thai then to be a translating expert to help us to get through with this. So translation is taking something in a code or a language and converting it into something more understandable or something else. In this case, the code that was copied from DNA during transcription is translated into a protein. That's to put it very simply, Samuel. That doesn't answer your question completely. It's just so that you can understand the basics of the difference between these two. But as we're going to go along here, some of that is being tackled on the questions, so let's look at that as we go along. In this question, they say the base sequence in a portion of DNA template strand is the following. Just to make sure that we got the pen on. So the DNA template strand has the following code: TGA, GGA, CTT. That is, we just took one portion of the DNA, and that's what we see there. The first question, they say, write down the base sequence of the mRNA codons transcribed from the template strand of DNA. So there's your word coming in there, Samuel, transcribed. And in this case, they're talking about transcription. Where does this transcription take place? Just for an explanation, it takes place in the nucleoplasm. How does it take place? The first stage is obviously DNA must be replicating. And as we looked at those three diagrams in question 2, where the DNA unwound and it unzipped, as it unzips, normally a DNA's nucleotides would come in and take the place of the DNA that has been detached. But in this case, what happens is messenger RNA sneaks in and steals the code. In a good way, though, because we need this code. It's not that the messenger RNA is doing us down. We won't lose anything. Actually, we're gaining something. So messenger RNA comes in and it copies the code from one of these two strands. And that's what they're saying. Here's the template. So DNA acts as a template during transcription. And in this case, they don't want to know the process. They want to know, write down the base sequence of messenger RNA codons transcribed. Now, guys, when you do this, remember that you must always start from left to right. And you always keep your codes or your nitrogenous base in triplets as they are shown here. Always in base triplets. So in this particular case, we have T and the opposite at. This would be A. And for G, it will obviously be cute girl, so C. For A, normally in DNA, we said at. But if you learned your RNA properly, you'd know that there's no T in RNA. So therefore, the T is replaced with a U, uracil. So RNA has uracil and not thymine. So please be very careful when you are making this match. They want to know all, so here obviously it would be C, C, U, and here it would be G, A, A. Please understand the following. Lots of learners think that whenever A is supposed to be there, or whenever A is supposed to be there, they put U. That's not the case. Whenever T is supposed to be there, that is where uracil comes in, because RNA does not have thymine. It has uracil in its place. So be very careful with that. And by the way, Samuel, that answered your question on transcription. So we've covered the transcription part. Let's move further first. Where in the cell does a process of translation occur? I'd answered that already for you. This takes place in the nucleus, more specifically in the nucleoplasm. We're still with the same question. Now, remember, we still have this here with us, because this is part of the question. Now look at this question. It says, use the information provided in the following table below to predict the sequence of amino acids synthesized from the DNA template strand. Now there's a DNA template strand here. The messenger RNA, just to remind ourselves again, was A, C, U. C, C, U, and G, A, A. This was the messenger RNA, and this was synthesized during transcription. Okay? But now look what they're talking about. And Samuel, you need to pay attention, especially here. Because now they're saying the table shows you the amino acids. Look at this table. It says the relationship between transfer RNA. Now they're talking about transfer RNA. We only spoke about messenger RNA. We didn't talk about transfer RNA. Now they're talking about transfer RNA, which are the anticodons. And here they're saying, in this particular story, they're saying here, these are the transfer RNA anticodons, and these are the amino acids that they carry. Now, Samuel, just to go back to your story, translation, first of all, takes place in the cytoplasm. It's the process where transfer RNA picks up amino acids from the cytoplasm and brings them to the ribosomes, where messenger RNA is stationed, uses the code provided by messenger RNA to combine, because the one would be the codon, messenger RNA, and transfer RNA would be the opposite, would be the anticodon. But each transfer RNA picks up a different amino acids. When you want to go home from Johannesburg or from Rudderport or from a shopping mall, you take different taxis or different trains. You can't get onto any taxi or any train. You'd probably get lost. You wouldn't know where to go. The specific taxis go to specific areas. Specific train numbers or bus numbers will take you to a specific area. The same way, a specific transfer RNA, we can consider transfer RNA to be the taxi for the amino acids. So different amino acids get onto the transfer RNA, but they all go to the similar destination, that is the ribosome. But each taxi can hook up, or each transfer RNA can hook up according to the code. Now here they're telling you that the anticodon for transfer RNA UUU will carry lysine. CGA will carry alanine, and so on. The whole table goes through there. Now we've got to go back to our story here. If this is the DNA and this is the messenger RNA, then what is the transfer RNA for this? We must understand something very easy here. Let me do that for you quickly. If we have a DNA here, and here we have mRNA, and here we have transfer RNA. These two guys are both opposite to messenger RNA. They are both mirror images of messenger RNA. So they would be very similar in their makeup. However, one golden rule is that RNA, no T, only U in its place. So for wherever T is supposed to be on an RNA, there would be U. So that's the clue that we must think about when we are going through this question. Let's go back to our template again. So if this is our DNA and this is the RNA, I said that the DNA and the transfer RNA are the same in most aspects. So let's draw it that way. If that's the messenger RNA, here's the DNA here, right? I'm going to rewrite all those letters. T, GA, GGA, and CTT. That is what the DNA has. But we know that RNA does not have T. So we take away the T there, we take away the T's there. So wherever we've taken the T away, we replace it with U. So that's a U, that's a U, that's a U. So that is how the transfer RNA looks. Now we want to know what is the sequence of the amino acids. Now let's look at it. UGA, you go to your table, look for UGA, that's one. Let's go back up. Next one is GGA, GGA, and that is number 2. And number 3 is CUU. CUU, that is number 3. Please understand, guys, that you do not have to memorize this table here. This table, if it's ever asked, will always be provided in the question paper. You do not have to know which transfer RNA picks up which amino acid. That is not part of your curriculum. You are required to know what is DNA, what is RNA, what is the difference, and how does messenger RNA form, and what codes will follow. So here, you look at it, so you will write it down in that order. 1 would be threonine, 2 would be proline, and the last one, 3, would be glutamate. Those were the 3 amino acids that would have been picked up. That's what you needed to do in this question. So please look at all the thinking processes that we had to follow. First of all, we know that DNA is the template. Secondly, we know that messenger RNA would be the opposite to the DNA. Thirdly, we know that transfer RNA has to be the opposite of the messenger RNA. Therefore, if DNA is the opposite of messenger RNA, and transfer RNA is the opposite of messenger RNA, it would be logical to say then, guys, that transfer RNA and DNA would be the same, except for there's no thymine in transfer RNA. Wherever the thymine is in the DNA, that must be changed to uracil in the transfer RNA. And that's the easiest ways, guys, to work out this situation. Again, you go through all those processes and you come to this particular answer. I'm hoping, Samuel, while we're talking about this, that your answer is being also answered. Because this what we are talking about is actually translation. When these amino acids now, just to follow through, when these amino acids are assembled now next to each other, they are assembled in a specific sequence according to the code provided, as we've seen here. This will be amino acid 1, amino acid 2, amino acid 3. These 3 amino acids are right next to each other. So the taxi brought the customers there. What the customers do is not the business of the taxi now. Transfer RNA's job is done. What was its job? Bring the amino acid to the ribosome. And immediately it leaves the amino acid there, goes away. But the amino acids are next to each other. And they make bonds between each other. 1 with 2, 2 with 3, and so on. And the bond that's between 2 amino acids is called a peptide bond. Okay? And this bond is created when the amino acids together lose 1 molecule of water, known as condensation or dehydration synthesis. They lose 1 molecule of water and they form. In this way, the different amino acids will make different proteins. And obviously, guys, we need 50 or more amino acids to make a protein, first of all. And this is the process of translation to actually answer Samuel's question.
Right, Mr. Aslam, I think we could take a quick ad break for these guys.
Definitely, they need it after that.
Yeah, it seems like a lot of information. So guys, I know that was very intense, but guys, remember, I always say, have a notepad out in front of you so you can just jot down notes so it helps you guys out. And guys, I'm loving what's happening on the page, more and more posts. That's always awesome for me. And guys, I can't stress enough, keep posting. And call in. I have posted the number on the page on our daily updates. So guys, make sure you get the number and get calling. But for now, we're going to take an ad break. See you soon. Welcome back, Mindsetters. I hope you had a nice little break. As I always say, make sure, guys, I can't stress this enough. Have a notepad out in front of you. This is revision, so make sure you guys are making notes because it's important, because without notes and revision, you're not going to pass the exam. And yeah, I actually found out Bonga and Samuel kind of have the same issue. So I think, Mr. Aslam, this next question should cover translation. And
yes, definitely.
All right, no problem. So, Mr. Aslam, take it away.
Thanks, Ty. And guys, we're back, and we still on protein synthesis. The question says the diagram below represents a part of the process of protein synthesis, and they give you a nice diagram. You look at the diagram, and the first question says, name the part or stage of protein synthesis that is illustrated in this diagram, or the diagram above. And obviously, Samuel and Bono, if you look at this diagram, you can see very clearly, there's your transfer RNAs coming, and this is the ribosome. Remember, you have this double semicircular type of structure that represents the ribosome. So this would be translation. So that's the first answer there. This is translation. And look at this diagram carefully because it's showing you what is actually happening. Here's the messenger RNA at the bottom there, messenger RNA there at the bottom, lined up on the ribosome, and you'll notice the transfer RNAs coming through with amino acids. Each one has an amino acid. Now you can see that they will combine according to the code, and when these amino acids are next to each other, they form those links that I was talking about. Sorry about my my line a bit off there. There, they make the link. That is what we call the peptide link. I'm glad this question came in correspondence to the question that was asked, so it helps. So these are the different amino acids. They are formed by peptide link, and every time two amino acid molecules join, water, H2O molecule is given off at each link there. Dehydration synthesis. And when we have 1, 2, 3, 4. When we have 2, it's a dipeptide. When we have 3, it's a tripeptide. More than 3, we call a polypeptide. And more than 50, we need to make a protein. Obviously, guys, this you can see is like a recipe. If we take different cakes, for example, let's say we're making cupcakes. That's the easiest I can think about. And you're making cupcakes, but you want your cupcakes to be red. Then obviously you're going to add red coloring to your whole recipe. If you're making cupcakes that you want to make green, you will still use almost the same recipe, but instead of the red coloring, you're going to use green. Now the same way, here's a recipe. We're using amino acids. There's only plus minus. There's debate on this, but for our syllabus, we talk about 20 different amino acids in almost all living organisms. And these 20 amino acids combine in a variety of ways to give us a variety of structures. Look around you. Look at your own family. You come from the same mother or father, or mother and father, sorry, I should say, mother and father, yet you look similar but subtly different, so that your parents can recognize each of you, unless, of course, you are identical twins. In that case, it's a different story. But other than that, we look different from each other. And in our bodies, our different parts, our hair, nose, ears, eyes, have different colors, have different textures, all because these 20 amino acids combine in different sequence. And where does this come from? The code provided by DNA, which is made up of smaller units, those base triplets, which we call genes. And these genes then will control the messenger RNA. The messenger RNA will control the amino acids from the transfer RNA, and this in turn will control the type, structure, and function of the protein that they are making. And this is the process of translation. Remember, the mark was only one. What I told you was an explanation to help Samuel and Bono. Name the organelle labeled W. And we've spoken about that already. W is the ribosome. Describe how the above stage takes place. And Samuel and Bono, now you listen carefully. Listen and watch. How does this stage take place? Remember, guys, and here is important rule of selection in your answer. One could go and explain the whole protein synthesis here. That's not what they're asking you. They're asking you about this stage. And this stage is known as translation. Because they haven't named the phase, you write it first down there, translation. And what happens here? First of all, we start with messenger RNA, which attaches to the ribosome in such a way that the bases are exposed. And which bases we're talking about? The nitrogenous bases. So the RNA is situated on a ribosome in such a way, this messenger RNA, that is exposing the nitrogenous base. And we call this messenger RNA the codon, because it's providing the code, which it stole from DNA. Stole in inverted commas, obviously. When this happens, the different transfer RNA molecules, they pick up amino acids, and they bring it to the ribosome. How? According to the code. Which code? Provided by the codon. So the transfer RNA is called the anticodon. The amino acids then link by dehydration synthesis. Are you with me, Samuel and Bono? I'm hoping you're with me as I'm giving to you one step at a time. The first step being the messenger RNA that attached to the ribosome. The second step being the transfer RNA picking up these amino acids all over the cytoplasm and bringing these amino acids to the ribosome. When these amino acids are next to each other, they link up by dehydration synthesis and form dipeptide, I would say first, and the peptide links, and they make polypeptides, and eventually they will make proteins. By the way, the transfer RNA goes away to pick up more amino acids. Again, like we said in DNA replication, we add a last bullet, all above controlled by enzymes. And that, guys, in a nutshell, is what translation is all about. And this question asks you that particular topic. So you need to know that as you're going along. To move further, still the same idea. The diagram below shows part of a messenger RNA. There they're giving it to you, messenger RNA. This one here, guys, I found very strange when we had it in the exams. Many learners couldn't answer the first question. And look how easy this question is. Really. It says, how many codons are shown in the diagram of this section of messenger RNA? The examiners made it easy for you to even number them for you. 1, 2, 3. And the answer, yes, you got a lollipop for guessing. 3. There were 3 codons here. Remember, the codons are made up of 3 nitrogenous bases, 3 nitrogenous bases, 3 nitrogen base triplets. Keep that in your mind. Base triplets all the time. They'll always be in 3s. And every 3 is 1. So, and they've also indicated for you in this diagram. Carrying on with this question. Write down the complementary base sequence of the DNA strand that formed codon 1 of the messenger RNA. And again, instructions, guys. They're asking you only for codon 1. So you go to codon 1, that's you. Where did it come from? From the DNA. The DNA that made this messenger RNA, what would have made you? Now, most learners would write here T. And that's where they go wrong. Because they think that where there's a U, there must be a T. But they're not asking you about the DNA. They're asking you about where did this messenger RNA come from. So therefore, we have to take that one out there. So this is a U. It came from an A. A would be the opposite of U in DNA. And U, the opposite of A, sorry, if this is messenger RNA, the opposite of A, at. That would be T, because this is DNA, and DNA has thymine. It's not RNA. They're asking you for the DNA here. And C would be G. And that would be your answer for 422, ATG. Please watch this mistake. Because of this idea that comes across that whenever there's a U, there should be a T, we try to make those mistakes. Okay? Lastly, explain the purpose of a specific sequence of codons in the messenger RNA molecules. Now let's understand this question. The language is loaded. It says explain the purpose of a specific sequence of codons. In other words, what they're saying, guys, is why are the messenger RNA sequenced in this way? What's important about this? Obviously, from what we've been talking about earlier, the messenger RNA would be coded according to the genes provided by the DNA. This, in turn, codes for different transfer RNAs to come and attach to the messenger RNA. Therefore, and we said that transfer RNAs are specific. They carry specific amino acids. And therefore, the specific coding would then specifically code different amino acids in a particular sequence. And this will determine the structure and the function of the specific protein that this messenger RNA is coding for. Very important there. Transfer RNA molecule carries complementary bases for a particular codon. Write down the complementary base sequence of a transfer RNA codon 1 in the messenger RNA sequence shown in above. There, we're going back. If this is U, then obviously this will be A. If that's A, this will be U, because now they're talking about transfer RNA, not DNA. So the U comes in there, and the last one is G. So this one will be there. Remember earlier, we spoke about ATG, because that was DNA. This is AUG, because this is RNA. And write down the, okay, B. What happened to B? B? Briefly describe the role of transfer RNA molecules in the translation process of protein synthesis. What is the function of transfer RNA? Do you have to, in this question, explain the whole of protein synthesis or translation? No. They only want to know from you, what do transfer RNA molecules do? What do they do? Transfer RNA molecules simply pick up amino acids from the amino acid pool in the cytoplasm and bring them to the ribosome according to the code provided by messenger RNA. That's all you needed to say there. Transfer RNA molecules pick up amino acids from the amino acid pool in the cytoplasm and bring them to the ribosome according to the code provided by the messenger RNA. If you want to, you can use the words codon and anticodon in your answer. In other words, transfer RNA acts as an anticodon, picks up amino acids, brings them to the ribosome according to the code provided by messenger RNA, which is the codon. That is your answer for that one. Describe the effect or impact that gene mutations might have on the formation of proteins. Firstly, obviously, guys, we need to understand what is a gene mutation. A gene mutation. Two words, gene mutation. A mutation is any change, in this case here, in the DNA or in the gene. This is not the same as we will talk about in meiosis, a chromosomal aberration or chromosomal mutations. That's different. Here we're talking about changes in a nitrogenous base sequence, an addition, a deletion, a swap, two bases swap for some reason, etc. So a gene mutation. They want to know if something happens to the DNA. Here the DNA, let's say this is the DNA, and the code was GCATATGAG. If that was the code. And for some reason, this code changed. What is the impact on the formation of proteins? This is what they want to know. And that's simple, guys. Obviously, if the DNA changes, if this A, for example, if this was the DNA, as we spoke about earlier, if that was the A, then obviously you would have got a U there originally. But if this A changes to a T, I'm doing it on there deliberately, if that changed, that's a mutation, because something has changed on the DNA. Then obviously this U can't be a U anymore, this U becomes an A. So obviously a change in the DNA or a change in the gene will affect the type of messenger RNA forming, and the type of messenger RNA formed will affect the type of RNAs that bring amino acids there. This in turn will affect the sequence of the amino acids, and therefore will affect the structure and function and the type of protein that is formed. So start at the top. The DNA changes, it causes a change in the messenger RNA. This in turn causes a change in the transfer RNA, which in turn causes a change in the sequence of amino acids, which in turn will result in a different protein being formed with different structure and functions. Ready?
Right, Mr. Aslam, I think we should give them another break because that was a lot of information.
Definitely, Ty.
All right, so guys, you know the drill. Keep posting on the page. I want to send a quick shout out to my fellas, Samuel, Skills, and Langa. Guys, I'm really loving the work you guys are doing, helping out all the other students. Guys, that's awesome. And Kashifa, as always, hi, how you doing? But for now, we're going to take a quick break, and we're going to see you after this. Welcome back, Mindsetters. I hope you had a nice little break. Guys, I cannot stress enough. Guys, keep posting your questions. I'm really loving what's happening right now. Like, all you have been posting, you guys have been chatting to each other, which is awesome. That's why this platform is there for you guys. So guys, I stress again, tell your friends to tell a friend, to tell a friend, to tell their friends to get on the page and get chatting, because it helps all of you, because we are trying to get that matric pass rate a lot higher. So guys, it only helps if you guys chat to each other. The more people, the better. But for now, before I link over, guys, make sure, we also want you guys to keep calling in. So make sure, 086-105-8262. Get chatting to us, call us, let us know what your questions are, and guys, we will tackle them for you. And right now, I'm going to hand over to Mr. Azza. Oh, wait, again. Before I forget, my mind is like everywhere, because I'm like reading all your posts at the same time. You know me, I'm trying to juggle things. But actually, one of the girls had a question here. Hold on, let me just load it up quickly. Because she wanted to find out, let me see if my computer decides to cooperate, because sometimes technology, it just doesn't want. Okay, let me see if it will pop up. Okay, in fact, no, when we go further on, I think I'll then just ask it there. Then we can just cover a question for you.
All
right, awesome.
Just keep it ready there. Yeah. Okay, guys, welcome back again. And we are now dealing with the next topic, and that is meiosis. Fairly easy. Learners normally do well in this section. It's about identifying the different phases of meiosis. Meiosis 1, meiosis 2, what is happening, what occurs throughout, what happens to the chromosomes, etc. This particular question gives you a diagram, and guys, this is how this question is asked. Generally, unless they ask you for differences between mitosis and meiosis, or sometimes they can ask you to explain the process of meiosis in an essay, but generally in section B, this is the type of question to expect. A diagram comes across, and they say diagram representing a phase of meiosis. In this one here, they say provide labels for parts A and B. Very simple. Just make sure that I got the highlighter on. A, they're referring to the structure here, and B, the structure here. What you need to be very careful about in this case is look at the structure carefully and see whether it's single-stranded or double-stranded. And in this case, if my eyes are correct, this is a single-stranded structure, and therefore A would be a chromatid, or chromatid, as some people may say. Chromatid, okay? That's A. And B is obvious, that is the spindle fiber. Still with that? How many chromosomes will there be in each cell formed from the cell at the end of meiosis? Now, this is also a famous question. They like to ask this. They give you a diagram like this. You can see the number of chromosomes here, and they ask you at the end of meiosis, how many? The trick is, you need to know whether this is meiosis 1 or meiosis 2. A simple rule of thumb to learn that, in meiosis 1, the chromosomes are lying together. The homologous chromosomes are lying together, especially in the first 2 phases. Remember, in anaphase, they separate. Also note, for your examinations, you do not need to know the names of the phases. But for logic, for easy flow, it's obvious that you need to know the phases so that you know what is happening. There's a progression as you're going along. But you do not need to name them in the exam. You'll notice, since last year, the question on name the phase has been left out. They only need you to know which is meiosis 1 and meiosis 2. So I was saying, meiosis 1, chromosomes are together. Meiosis 2, chromosomes are separate. They are single. And here's a classic one. In anaphase, this is a classic one. In anaphase of meiosis 1, this situation will not occur because this would be that. You'll see two strands separating. A whole chromosome would be separating on either side. Here you can see chromatids are being separated. So obviously in this particular, in this case here, you've got 1, 2. And these are going that way, and these two will go on the other side. So the end of meiosis, in this case here, we would have 2 chromosomes. In meiosis 2, I said the chromosomes lie singly. There you have it there already. So in other words, the number of chromosomes in each cell is shown in this diagram here already. There are 2. Now many learners would say 1, 2, 3, 4. And that's where they go wrong. Because there are only 2 chromosomes altogether here. And the chromatids have separated. So it'll be 2. How many chromosomes were present in the phase before the one represented in the diagram? Before this separation took place, how many chromosomes were there? Now this is basically a trick question. Because you're going to think, because I answered 2 earlier, now I must say 4. They want to know before this phase here. They didn't say meiosis 1, meiosis 2, but before this phase took place. How many chromosomes? In other words, metaphase in this case here. How many chromosomes would there be? There would still be 2. Because even though you're seeing 4 strands here, there are 4 strands, if you put them together, 2 chromosomes. Because 1 chromosome is made up of 2 chromatids. So therefore, before this also there would be 2. So be very careful there. Name 2 reasons why meiosis is biologically important. Why does meiosis take place, in other words? What is the purpose? What is the significance? Lots of different words they can use there, all mean the same thing. In other words, why is this process taking place? What is its purpose? And meiosis, obviously, is there to make sure that the chromosome number remains constant from generation to generation. Let's take humans, for example. We have 46 chromosomes in our normal body cells, which we call the autosomes. Those chromosomes that are found in the ordinary body cells or somatic cells. Ordinary body cells are called somatic cells, and the chromosomes that are found in there are called autosomes. And we have 46 in each of our cells. In other words, if I take a piece of my nail and we put it under a microscope, we will find 46 chromosomes there. If I take a piece of my eyelashes or eyebrows, and we can find the same there. If I take a piece of my skin, we're going to find the same thing there. Now, meiosis makes sure that from our predecessors, from our parents, coming to us, they had 46, we still have 46. You must remember that for us to be conceived, fertilization had to take place. There was some fusion. Now, if we had 46 from the mother, 46 from the father, then obviously we would be having 92. And our children would have 184, and so on, and so on, and so on. But that doesn't happen. Because when sex cells or gametes are produced, then the number of chromosomes is reduced by half because of meiosis. So it makes sure that the chromosome number stays the same. One important process you learned in meiosis was crossing over. What is crossing over? Is the exchange of genetic material from one chromosome to the other. As you can see, this coloring here, if we went further in this diagram earlier, prophase, we would have seen that maybe this particular chromosome was one was all white and one was all black, and because of the crossing over, they've changed their structure here in some way. What is this crossing over? It's the exchange of genes. And what does this do? It brings about a variety. That is why all the brothers and sisters in a family do not look exactly the same. They may have features which are stronger from the father or the mother, but each one is an individual and each one is unique in different ways. Why? Because of their specific genetic makeup. And meiosis brings about this difference. Otherwise, imagine if we all believe that we all came from Adam and Eve, then you, me, and everybody, Ty sitting next to me, we would all look the same as Adam and Eve. And I would be saying, Adam, how are you, Adam? And Ty would be saying, Adam, how are you, Adam? And so on. Imagine a cop and a criminal. I'm the detective, my name is Adam. The criminal, his name is also Adam. And I want to take his fingerprints, Ty.
That would get very, very confusing very quickly.
And messy.
Exactly.
Okay, so meiosis makes sure that this doesn't happen. We all look uniquely different. So those are the important. And also, when you do reproduction in paper 2, you will talk about alternation of generations in mosses and in ferns. Meiosis makes that possible. Meiosis makes alternation of generations in plants possible. Those are the significance of meiosis, or the importances of meiosis. Moving on, tabulate 4 differences between meiosis and mitosis. I told you this at the beginning, that you need to know the differences between meiosis and mitosis, and this is the time where you should be scratching your head and thinking in grade 10, oops, that was a long time. For some of us longer, for some of us shorter, we know. So we need to know the differences. And the important thing, instructions again. What did they say? Tabulate. Normally, and here you have it. How many marks here? 9 marks. And there's 4 differences. 1 mark for each difference should give you 8 marks. But they've given you 9 marks. Why have they given you 9? Because you had to tabulate. And they're giving you 1 mark for drawing the table. Obviously, you will draw the table, and you'll put there mitosis, meiosis, and you would tabulate the differences. First of all, a meiosis takes place for the production of somatic cells. And meiosis for the production of gametes or sex cells. Please understand, lots of textbooks and learners, they say that mitosis takes place in somatic cells, which is correct. That's fine. And then they go to say that meiosis takes place in sex cells. That is incorrect. Meiosis does not take place in the sex cell. The result of meiosis is a sex cell. So meiosis actually takes place in the sex organs for the production of sex cells. Okay? That's the difference. That is why we've highlighted here for the production of somatic cells and for the production of sex cells there. And the mitosis obviously is exact duplication. That means cells are identical. In meiosis, cells formed are different. The chromosome number here would remain the same. And here the chromosome number is halved from diploid to haploid. The mitosis is 1 division, meiosis 2 divisions. You can add in mitosis 2 cells form and meiosis 4 cells form. That is it. In your case, you would also be required to rule lines across that way so your table is complete as you're going along, because that's what you're getting marks for. So make sure that you have understood that and you know that you put in your marks accordingly. Also, guys, when you're drawing your table, you cannot write here somatic cells here and then write here 4 cells next to it. You've got to compare what you are comparing. They must be corresponding to each other. If you said that, you will have your somatic cells here, and on the other side you're going to have a problem in that you're going to get you won't get a mark on that side. In other words, we'll give you a tick here, and if you wrote here 4 cells, that one will be marked wrong. So please make sure that when you're drawing your tables, 1, you completely draw all the columns and all the rows. Secondly, make sure that you compare things in the same row. What you are talking about, the same feature you're talking about must be in the same row. You cannot mix them up. Don't write down one list on the left-hand side and then write another random list. There are differences, but they are not differences at that particular topic. Please make sure that you keep that, otherwise you lose marks unnecessarily. Okay. The last question in this section. Polyploidy refers to an organism, usually a plant, which has more than the diploid set of chromosomes in the nucleus. Example, triploid plants have 3 sets of chromosomes, and tetraploids have 4 sets of chromosomes, and polyploidy many. In this case, it's saying it's rare in animals, but more common in plants. First question says, briefly explain how cells may become polyploid. Okay, cells become polyploid. Now here, we must talk about meiosis. During meiosis, chromosomes do not, the whole set of chromosomes do not separate. And this normally, obviously, if you know your meiosis, it takes place during anaphase 1 or anaphase 2, when the chromosomes do not separate. In other words, what would happen then, one gamete would have double, or rather diploid, chromosome number. If this gamete now fuses with a haploid, then 2 plus 1 equals 3. It will form a 3N or triploid. Okay, so if that happens and if it doubles, then you'll have more. If 2 and 2 becomes 4 and so on, and that is how this thing happens. We'll just stop there and I'll hand over to Ty.
All right, guys, you know what? I need you guys to make sure you stick around, yeah? Promise, you're going to stick around and make sure that you guys, keep posting, keep chatting to each other. Make sure that you guys keep chatting, because that's why the platform is there for you. But guys, this is where I sign off and I say, see you next time.